Good afternoon, everyone, and welcome. I'm Mac Titchener, president of the Osteosarcoma Institute, and along with our friends here at Old Parkland, we're very excited and pleased to bring you this program tonight. In the next hour, we'll witness how science synthesizes discoveries from disparate areas, disparate fields, and makes progress by combining sometimes unrelated, seemingly unrelated discoveries to expand or sometimes completely change our understanding. We'll also see how these gains in understanding can change the paradigm for advancements, in this particular case in the field of medicine. The Osteosarcoma Institute deals in one small corner of that field, a bone cancer called osteosarcoma. And that osteosarcoma happens to play a part in this particular story you'll hear tonight. Our mission is to facilitate progress against this very difficult disease. We're based here at Old Parkland, but international in scope and supported by a distinguished scientific advisory board of physicians and researchers from the leading institutions in the country, many of whom are here tonight. We appreciate them being here and appreciate their hard work. Through a late-breaking, extenuating circumstance, our director, the Institute director, Dr. Lee Hellman, cannot be with us tonight, but he is going to join us electronically if all goes right. His bio is in the program, as all the speakers are, but he has a distinguished academic background, various leadership roles at the National Cancer Institute and Children's Hospital of Los Angeles, and has treated pediatric cancer patients for many years. And Lee?
Thank you, Mac. Can everyone hear me? Am I audible? Okay. So it's a pleasure to be here, unfortunately, only by Zoom. My wife came down with COVID yesterday, and I thought it was a better part of valor to not get on an airplane and potentially expose colleagues and guests. I started treating my first patient with osteosarcoma in 1983. It's a long time ago. And in fact, we were entering patients on a trial that was the first trial that went on that made a dent in osteosarcoma, and it proved that chemotherapy prolonged and cured patients at a much higher rate when every patient with localized disease was given chemotherapy. But since that time, there's been precious little progress. And I am thrilled to be part of the Osteosarcoma Institute. We will not let this fail. I appreciate each of you for coming. And I just want to say, part of what you'll hear is how evolution and cancer go hand in hand. And what we're beginning to understand is the more we learn about evolution, the better we will understand cancer, and not just osteosarcoma, but many, many cancers that are recalcitrant to current therapy. So with that, I'm going to stay on and listen, and I'm thrilled that this event is happening, and I thank all of you for coming. And Mac, I'll turn it back over to you.
Thank you, Lee. It's now my pleasure to introduce the moderator of our panel tonight, Dr. Michael Brown. As many of you know, Mike wasn't born in Texas and technically didn't get here as quick as he could either. Got his medical degree at Penn, did a residency at Mass General, and postdoctoral work at the National Institutes of Health. But he did get here to UT Southwestern in 1971, and with his colleague, Dr. Joe Goldstein, discovered the low-density lipoprotein, or LDL, receptor which controls cholesterol in our bodies. Their work laid the groundwork for development of the statins, which many of you know, some of you take, to lower cholesterol and prevent heart attacks. And of course, it's made a huge positive impact in the health of many people. Dr. Brown and Dr. Goldstein shared the many awards for that work, including the Nobel Prize in Medicine and Physiology. So, Dr. Brown, thank you, and over to you.
Well, thank you, Mac. Thank you for founding the Osteosarcoma Institute, and thank you for sponsoring this symposium. So our topic is osteosarcoma, a cancer that kills young people, but in a larger sense, we'll talk about the cancer process itself. You'll hear that cancer is as old as animal life. Even dinosaurs had cancer. And you'll hear from experts who will explain how cancer arises from mutations in genes. And we will end on a ray of hope that science will eventually win the battle against these horrible diseases. So why do we have cancer? The answer lies in the beginning of life. Each of us began as a single cell, a tiny fertilized egg. The egg divided into 2 cells, and the 2 became 4, and then 8, and then 16. After 9 months, your body contained 1 trillion cells. That's a 1 with 12 zeros. And some are skin cells, liver cells, brain cells, and bone cells. How can a single cell become 1 trillion different cells all in 9 months? The answer is genes, the instruction book for building a human body. The fertilized egg has 20,000 genes written in DNA and organized into 46 chromosomes. Each chromosome is linear like a string of beads. Imagine 4 colors of beads, red, blue, green, and orange, arranged in a sequence along an extremely long strand. Each cell contains 6 million beads in a precise order. When a cell divides, the beads must be copied so that each daughter cell inherits the same sequence. 6 million beads, that's the number of letters in 1,000 copies of the King James Bible. Every time a cell divides, it must copy 1,000 Bibles. And billions of cells are dividing in our bodies every day. Copying isn't perfect. Mistakes occur. A blue bead is inserted where a red one should be. We call that a mutation. Most mutations kill the cell, but that's okay. We've got trillions of them. But other mutations are serious. They cause a cell to keep dividing even when the body plan calls for division to stop. Or they cause a cell to migrate to a place where it shouldn't go. Or a mutation can cause a cell to refuse to die on command. The result is a cell that keeps dividing, moves to the wrong place, and refuses to die. We call that cancer. I believe that cancer is unavoidable because cancer and evolution are 2 sides of the same coin. Errors in DNA copying cause cancer, but when they occur in eggs or sperm, they create new species. Mutations caused a paw to evolve into a hand. If mutations didn't occur, then life could not have evolved beyond the tiny microbes that first inhabited the earth. Fortunately, evolution has given humans the brains to understand mutations, and we're learning fast. When I graduated from medical school, dare I say, a half-century ago, we knew nothing about genes. We didn't even know how many chromosomes we had. We knew nothing about mutations that cause cancer. Now we're unmasking cancers one at a time, and cures are certain to follow. Now I'd like to introduce our first speaker, Dr. Alexandra Callan. We haven't time for formal introductions, so I refer you to the brochure that you received. But I'll say only that Dr. Callan is a gifted surgeon and is director of orthopedic oncology at UT Southwestern Medical Center. She has dedicated her life to the care of patients with osteosarcoma and other cancers of bone and soft tissues. Dr. Callan, please tell us about your patient.
Thank you so much. Good evening. It's a treat to be here with you all in this beautiful space. And it's now my honor to be able to share with you the story of a brave young girl who was my patient, and her name was Izzy. At 10 years old, Izzy was an incredible dancer, and she had recently competed in a national dance competition. 2 weeks later, in the middle of one of her dance lessons, she felt her right leg snap beneath her as she crumbled to the floor. She was brought to a local emergency room where x-rays pictured here revealed a pathologic fracture. A pathologic fracture is a broken bone that occurs when the bone is weakened by tumor or another disease through a low-energy mechanism. This means that the bone breaks without normal trauma, such as shifting your weight during a dance move in ballet practice. So the arrow there is pointing to her fracture. This is the end of the femur or the thigh bone. And you can see all the signs of an aggressive bone lesion or an aggressive cancer. There's bony destruction and extra bone deposition, which we call osteoid matrix. There is elevation of the periosteum, which is a sign that the cancer is growing very quickly and elevating that surrounding membrane around the bone. In the middle of the night, Izzy was transferred to Children's Medical Center in Dallas, where I met her and her family and performed an open biopsy and placed a cast for her broken leg. The biopsy confirmed every parent's worst nightmare. Their daughter had bone cancer, specifically osteosarcoma. We completed a cancer staging workup, which involves a CAT scan of the lungs, a whole body bone scan, and an MRI of the entire femur. Thankfully, she didn't have any signs that her cancer had spread to any other locations, meaning that there was no evidence of metastatic disease. But the cancer filled her entire femur bone. So now the first battle with cancer begins. She was started on standard chemotherapy for osteosarcoma, a treatment regimen that has not changed in over 40 years. During this time, it gives me the opportunity to start planning surgery with a custom-designed implant specifically for Izzy. After 10 weeks of chemotherapy, she is ready for surgery. It was my job to remove the entire femur. You can see a model of it pictured here. And then I replace it with a gigantic joint replacement. In the x-rays there, you can see a total femur replacement involves a partial hip replacement and then a total knee replacement. This special implant designed just for Izzy has a magnet inside that allows me to grow her right leg as her left leg continues to grow. When the pathology results came back, I had negative margins, which means I removed all the cancer from her body, and the analysis showed there was 98% necrosis. Hooray, that means that the cancer was killed by the chemotherapy, and now we were on the last phase of treatment. So even after surgery, patients undergo another 20 weeks of chemotherapy to ensure that we eradicate any hidden tumor cells throughout the body. Izzy made it through the full treatment for osteosarcoma and was declared cancer-free. She rang the bell, she had her chemo port removed, and we continued to lengthen her prosthetic femur using an in-office magnet to allow her right leg to grow as she grew taller. Sadly, only 4 months after being declared cancer-free, she started having pain in her left jaw. Imaging revealed that her cancer had now come back. You can see that pictured in the skull model here. Now the second battle with cancer begins. Izzy now had metastatic osteosarcoma, meaning that the cancer once found in her leg recurred in a distant bone, specifically her jaw. This is devastating for the patient, the family, and her doctors. I fought to find a new surgical team. She endured another major surgery to remove the tumor in her jaw and skull, with a reconstruction utilizing the muscle from her left thigh. She again started intensive chemotherapy and required radiation to the spots in the skull that could not be surgically removed. Unfortunately, the cancer continued to progress. She went through several regimens of additional high-dose chemotherapy and even started a clinical trial that utilized her body's own immune system to try to fight the cancer. She underwent more radiation. The disease progressed to her lungs and continued to grow near her eye and her brain. She continued to fight her osteosarcoma battle with bravery, hope, and grace. And after 2.5 years of heroically fighting the cancer in her body, she died surrounded by the love of her family at home. In her short 12 years on this earth, she has made a greater impact on my career and in the fight for osteosarcoma than any other human. She raised nearly $500,000 for osteosarcoma research, and she touched the hearts and the lives of children and other families across the world. Izzy and her family connected me with the Osteosarcoma Institute. And just like me, that is why you are here today. To hear more from the brilliant scientists on the stage with me, to talk more about how we need to continue to look for innovative and revolutionary options to cure cancer, including osteosarcoma. And so now, it is my great pleasure to introduce Dr. Chand Khanna. Dr. Khanna is a distinguished veterinary oncologist, pathobiologist, and innovator. He is the Chief Science Officer with Ethos Veterinary Health and President of Ethos Discovery, a nonprofit incubator for scientific innovation. He is currently serving as the Strategic Advisory Board Chair here for the Osteosarcoma Institute. Dr. Khanna, please tell us more about osteosarcoma.
Thank you, Dr. Callan. So as you've heard, I'm a veterinarian who treats cancer in animals. So my patients are four-legged, furry creatures. The research I've done is to understand why the cancer that affects my patients, these pet animals, and Dr. Callan's patients, children with this disease, have the disease spread to other parts of the body, and how can we take that learning to develop new drugs to prevent that from happening. We are here today to use osteosarcoma as an example of thinking in a sort of a holistic setting, as Max suggested initially, and take a example from nature, which comes to us by the fact that osteosarcoma is a disease that's very old, nearly 2 million years ago, first found in a fossil from a hominin in South Africa, and as you'll learn soon, 70 million years ago in a dinosaur. So that knowledge about evolution and this cancer, we now seek to apply to the challenge that we haven't overcome, which is why this cancer is so difficult, why we can't do more to prevent the disease from spreading. So that starts with osteosarcoma as an example for how we can use evolution in a broader sense. And we'll learn more about evolution and hopefully bring that back to the research that we do in this cancer and other cancers in a more general setting. So to share a couple of highlights about osteosarcoma for background for you, it is a disease that occurs in the long bones of the body and seems to occur where bone rapidly grows. And in dogs, this is a disease that I see in my patients that are typically Irish Wolfhounds and Great Danes in their long bones of the body. So there's something about the growth of these bones, and you'll learn more about that in the dinosaur, that is connected to this disease and why it's so difficult. And we're going to take that knowledge to continue to work on how we develop new therapies for the disease. Important point for all of us is to consider the fact that, as Dr. Hellman said, in 1986, there was a significant advance through the addition of chemotherapy to patients. So surgery and then chemotherapy was 1986, significant advance in the care of patients at that time. And so well over 40 years later, really no significant change. So we have our work cut out for us today. And we're now looking outside of the box to look at evolution, to look at the dinosaur, and to understand how these genes that cause or allow evolution and cause cancer, how the learnings from both sides of this coin can help us tackle this important clinical problem. So I'm really excited to be here from that perspective and to take the field forward beyond the stagnation that's occurred for 40 years. And the last point is that in this disease, most commonly, it's the spread of the cells from the bone to the lung. And those cells, from discussions we've already had around this conversation about evolution, those cells become stressed, must adapt, and evolve to avoid the death from the stress that they're experiencing. So that's how we're trying to draw the line between evolution and cancer. And I'd like to now pass the ball to Dr. Evan, who is a dinosaur scientist. So the applause can begin for this. And Dr. Evans is from the Royal Ontario Museum in Toronto, Ontario, Canada. And that's important to me because that's my hometown. And I used to walk from my elementary school in grade 3 and 4 to his museum and got excited in science. So it's a great pleasure to introduce Dr. Evan.
Thank you, Dr. Canna. It's very exciting for me to be here today. I mean, if you asked me as a dinosaur-crazed 4-year-old, I never would have dreamt I'd be on stage with leading medical professionals discussing the future of cancer. But research can lead you into strange directions you wouldn't expect, and this is certainly one of them. I'm here today because my team diagnosed osteosarcoma in the shin bone of a 77-million-year-old horned dinosaur called Centrosaurus from Alberta, Canada. And this is a relatively new discovery. We just announced it less than 2 years ago during the pandemic. Now, Centrosaurus is a dinosaur that you might recognize. It's a close cousin of Triceratops, which is one of the most famous dinosaurs out there. As a horned dinosaur, it has the classic body plan of Triceratops, a big horned face with a neck shield, a rhinoceros-like body, and a long tail. Centrosaurus itself was smaller than its famous cousin. It was about the size of a white rhino. And it's differentiated by the arrangement of the sort of horns and frills on its skull. Unlike Triceratops, which has the 2 big horns over the eyes and the small one over the nose, Centrosaurus has the opposite configuration. It has this long rhinoceros-like horn over the nose and 2 small stubby horns over the eyes and a much more ornamented neck shield. The discovery of cancer in this dinosaur represented the first confirmed case of an aggressive cancer in a dinosaur and the second oldest case of bone cancer in the fossil record. The world of this unfortunate dinosaur was very, very different from it is today. Global temperatures were much higher than they are today, about 20 to 30 degrees Fahrenheit higher, and there were no polar ice caps. It was, in general, a warm, wet, lush world that supported a rich and diverse ecosystem of plants and animals, some of which were familiar to us today, but it also included a wide and spectacular array of dinosaurs. As living animals, these dinosaurs were susceptible to a wide range of diseases, including bone cancer, as my team recently showed. An exciting aspect of our research is that we approach the diagnosis in our dinosaur patient as we would in a human patient in the fossil record for the first time. We assembled the largest pathological team brought to bear on a fossil disease, which consisted of 8 medical professionals and 3 paleontological specialists. We employed a full set of diagnostic techniques from big to ultra small, as in human patients, using the gross morphology or anatomy, radiological examination, and right down to the bone cell by studying the fossil bone under the microscope. I actually have one of the thin sections we made of the original fossil right here. And this step was really essential to confirming the diagnosis. It's basically a biopsy of a dinosaur. And it allows us to study the cancerous tumor at the cellular level, which was critical to the final diagnosis. We compared our dinosaur tumor to a normal shin bone in the same species. If you look on the right, the far right-hand side of the screen there, you can see a normal shin bone. So a very simple columnar bone, and then right beside it, you can see the cancerous bone with the large tumor mass. And we also compared it to a human case in the exact same bone, the fibula or the shin bone. Now, most diseases expressly manifest in soft tissues like organs. So think lung cancer or pancreatic cancer. And the fossil record is dominated by hard tissues like bones and teeth. So studying any ancient disease is a real challenge, and this is particularly true of rare diseases like cancer. But bone cancer offers the rare ability to track cancer in deep time, even in a dinosaur. The dinosaur pathology itself consisted of an enlarged, gnarly, baseball-sized growth in the upper end of a shin bone, just as you would expect in a human. And in this case, bone malformation had extended all the way to the ankle of this individual. With CT scan imagery, as you can see here, we could visualize the tumor in 3D. Remarkably, we were able to trace the tumor development through the bone, and you can see our spinning visualization here, from the main tumor mass all the way to the ankle of this individual. And this allowed us to confirm this was osteosarcoma or bone cancer at a very advanced stage. This is the first cancer diagnosis in the fossil record done to a clinical medical standard, and very exciting result for scientists well beyond paleontologists like me, and I think the interest from the medical community has really been outstanding. What's more remarkable is that the manifestation of the bone cancer in our dinosaur is virtually identical to what we see in the human case, appearing in the knee end of a lower limb bone that's in a rapidly growing individual, the highest risk bracket for osteosarcoma in humans, as Dr. Callan discussed. The finding in a dinosaur suggests that bone cancer has a very deep evolutionary history, going back to the shared common ancestor of humans and dinosaurs. And that goes back 310 million years. And it likely goes back even further to the origins of bone itself. Not only does cancer in a Centrosaurus teach us about the evolution of cancer, it also provides us some unique insight into the biology of these horned dinosaurs. The advanced development of the tumor allows us to find cancer, the advanced development of the tumor allowed us to find this cancer among thousands of bones excavated from a bone bed containing a herd of hundreds of individuals of the same species, Centrosaurus. The advanced stage of the cancer would certainly have been very painful to this individual and limited its mobility for quite some time, and it's likely the cancer had metastasized by the time of death. Under normal circumstances, this would have made this individual very vulnerable to the top predators of the time, the famous tyrannosaurs. But protection from its herd, from its social group, and even its family allowed the animal to live as long as it could. And rather than passing from this horrible disease, it passed with the rest of its herd in a massive coastal flooding event. So our cancer patient had a dinosaur support system. So now I'm going to pass it to Dr. Paul Meltzer. He's a leader in the application of genome science to cancer research. Dr. Meltzer is broadly trained in clinical medicine and biological research with extensive laboratory experience in molecular biology. Dr. Meltzer was recruited to the National Cancer Institute as chief of the genetics branch, where he served for the last 15 years. His research is dedicated to the investigation of genomics of pediatric cancers and other rare tumors with unmet medical needs.
Okay. Well, I'd like to continue some of the themes that have already been started and tell you a little bit about the state of knowledge of how osteosarcomas develop from a genetic perspective. But I have to say before starting that as a youngster, I became fascinated with dinosaurs and regularly frequented the great fossil collection in the museum in my hometown. And eventually, though, I became even more fascinated by the discovery of the DNA double helix, which I don't know if you noticed, there happens to be a painting of that discovery just outside the door of this room. And that's the one that took hold, and I've ended up devoting my scientific life to the study of genetics and ended up focusing on how DNA relates to human health, particularly related to the development of cancers. And these are a group of diseases that can be characterized as diseases of the genome because they have errors or mutations in the normal human genome sequence. That's the sequence of all the chromosomes with the genes and everything that connects them and carries the signals for how the whole system works together. But never did I imagine that I would ever be a participant in a program where both human and dinosaur cancers were discussed at the same time. Nothing further from my mind. But unexpectedly, in recent years, we learned from Dr. Evans that dinosaurs can develop osteosarcoma. I was pretty happy to have learned that we could find it in ancient hominins and how it develops in other vertebrate animals. But it's really interesting to see that it goes this far back. So you have to ask the question, what do we have in common with dinosaurs other than being, we're definitely not usually as big as they are. But so, what do we have in common? First, as you can see from this slide, just looking at a dinosaur skeleton, you see features that look an awful lot like pieces of a human skeleton. So you get a sense that they're really kind of related things. So we have major similarities. And as Dr. Evans has pointed out, the rules for making and maintaining a bony skeleton are likely to be very similar and conserved over the millions and millions of years that separate us from the dinosaurs. That's pretty interesting because that means that these tumors develop in the context of really ancient biological pathways that go way back. And understanding those rules for maintaining normal bone and developing normal bone has to be relevant to the biology of a human osteosarcoma and could well be useful in guiding research aimed at helping our patients and possibly related to other cancers of different sorts. And you've already heard, but I'll reiterate that the human genome, which resides in the nucleus of every cell, contains billions of letters, G's, A's, T's, and C, encoding large numbers of genes, more than 20,000. But now, if I could have the next slide. So this slide is meant to convey the concept that in normal bone, the genes work together on the left, and you get a development of normal, healthy bone, which is an amazing tissue. It can grow into the correct shape and size. It can produce and remodel the mineralized calcium matrix, containing matrix that gives bone its strength to repair injuries. And in the end, it can appropriately interact with other kinds of cells that also occur in or near bone. So the normal bone that we and all other creatures with similar skeletons rely on, it's a wonder that has evolved over many million years, a very complex, wonderful, and fascinating tissue. Yeah. Well, now the question comes up, how does cancer arise? Every time a cell divides, as you've heard, it replicates perfectly, not quite, but and that's important and essential for evolution to occur in the natural world. But it does also open the door to cancers occurring. And if a very rare mutation occurs that disrupts processes central to regulating cell growth and division, unbridled cell proliferation can follow, and that's when you will start to see the development of a cancerous tumor. And many scientists for the last several decades have been working at identifying the specific mutations in many different kinds of cancer, and the technologies have evolved to an extremely high level right now so that we can learn a lot fairly quickly about any kind of cancer for which we can get appropriate specimens. And so finding those mutations and deciphering how they lead to cancer has become an important industry in molecular biology research. But for today, let's consider specifically what happens in osteosarcoma as an example showing how this kind of work can help us understand the genesis of cancer in general. Now, when we look at an osteosarcoma with the latest generation of tools for genome characterization, we find that it appears to start with a bang, a mutation in one cell in growing bone that destroys a single gene, most commonly the p53 gene, often called the guardian of the genome. This can be enough to start the growth and evolution of an osteosarcoma tumor. Without p53 to maintain the genome integrity, many other genes in that cell can go rogue, and that's what the right side of the slide is meant to convey. So accumulation of mutations, and there are many different kinds of mutation, but in the end, you end up with something that doesn't behave normally. Now, studies of lots of cancers have identified alterations in growth-regulating genes that are relatively easily targeted with specific drugs. And these have led to great advances in precision cancer medicine that really have been, in fact, impactful to some cancer patients. This has been a great thing, but unfortunately, this class of targets is very uncommon in osteosarcoma. Instead, we mainly find alteration in some of the core cell division machinery genes, which as yet are not quite so easily targeted. And as Drs. Hellman, Kana, and Callan have explained, what we mainly do now is treat patients with generic chemotherapy drugs that kill rapidly dividing cells in many different kinds of cancer. And what the real special thing is done by Dr. Callan, who and her colleagues who have over the last decades drastically improved surgery for these patients and made real progress in this disease, while those of us who are working on the medical side have seen very, very little progress by comparison. What we have done, again to reiterate, is that the use of chemotherapy and surgery does cure some patients, but not nearly enough. And we have not proceeded to major improvements for some decades, and it's clear that there's much that remains to be done. The next slide, please, Amy. So let's return to what is happening in the tumor. Okay, so once the cat is out of the bag, then and a good cell goes bad, then you have a situation where evolution again takes hold. So as that first cell divides and its progeny divide, there are opportunities and the certainty of new mutations occurring. And so you end up with a branching tree that looks very much like an evolutionary tree of coexisting, we would call them strains or clones of cells that are growing within the tumor. Now, those don't all necessarily behave identically, and they don't all need to respond equally well to the treatment that we give. And that opens the door for relapses, for the development of cell types that spread easily to other parts of the body and adapt, and creates a very significant challenge for patients, doctors, and researchers with this disease. So we start off with probably a more complex tumor than we have previously realized. So just as all living creatures are enmeshed in their habitat and adapt to habitat changes during evolution, cancer cells also are enmeshed in their bony habitat and can adapt to changes in their neighborhood. External forces like cancer treatments can lead to the selection of surviving cells that have advantageous changes to those tumors. And a promising treatment can fail because of this. This is one of the reasons that multiple anti-cancer agents are often used together. Just as in the evolution of living organisms, osteosarcoma cells that develop mutations favoring growth and/or spread of the tumor will tend to survive and become more difficult challenges to treatment. Next slide. When we look at newly diagnosed tumors, we find that they have already developed incredibly abnormal, highly evolved genomes, often containing hundreds of abnormalities. No two patients' osteosarcomas have identical changes in their genomes. In the illustration, this picture of circles illustrates the diversity of genomes because each one represents a different patient. There's no need to look at all the details, but the red arcs in each circle represent how DNA strands have been broken and erroneously rejoined. And I think you can get the impression that there are a lot of these, but that they're different in every patterns. This is an intimidating situation. Next slide. But just as snowflakes that have endlessly complex forms that arise from the fundamental rules guiding their formation, we have come to understand that osteosarcoma genomes follow a set of rules that lead to some common biological states. These rules, though hidden in the complexity of the disordered genomes, are beginning to be understood and have potential to unlock new opportunities for treatment for osteosarcoma and perhaps other cancers based on significant biological pathways that are important for tumor growth. Importantly, monitoring what is going on in an individual patient over time is becoming more practical, and it may be possible to individualize and modify treatment to account for the ongoing evolution of a tumor. Other approaches to treatment that are based on the essential wiring diagram of the osteosarcoma cell may perhaps evade some of these problems. And strategies for harnessing the power of the immune system are also on the research agenda. But I believe that tenacious scientists leveraging the current state of knowledge and the current advanced technology that are available, I feel confident that these studies will lead to progress in the treatment of osteosarcoma and very likely other cancers. So I now will pass this topic over to Damon Reed, a distinguished leader in osteosarcoma translational and clinical research. Dr. Reed is a pediatric oncologist at Moffitt Cancer Center and the clinical co-leader of the Evolutionary Therapy Center of Excellence at Moffitt. So really somebody who's been thinking about this problem of tumor evolution in a very practical context. So I'm very excited to hear what he has to say.
Thank you, Dr. Meltzer. So I'm one of the several 100,000 professionals in the country that tell people you have cancer. And sometimes it's to an adult, sometimes to a child and their family. Not super fun thing to do. But at the same time, we've heard about beads in DNA, we've heard about mutations, we've heard about a lot of different concepts of cancer. You've seen a bone that's, I can't even wrap my brain around 76 million years ago. It's so hard to, it's so abstract. But what is cancer? Like, what is it? And so I think a lot of people would think it is mutations. It's the same cell over and over again. It's a disease. It's otherworldly. It's gross, or it's I don't know, scary. It's all these sorts of things. I tell people I'm an evolutionary oncologist, and I think I'm the only one. And I think that's why I got invited to this prestigious panel. Just a Florida guy. And so, but what does that mean? What do I think cancer is? I think those thoughts are too static. I think those are make cancer too simple. I think cancer is fundamentally one cell in, let's say, my body that becomes a new species. I'm the world, and it's a new species, a new type of animal inside me. And that animal is inside me in a lush environment like Dr. Evans described. Warm, nutrients everywhere, great place to grow up. and it keeps growing and keeps growing, and it makes more cells. Those cells don't coexist like these friendly dinosaurs, but rather are fighting tooth and nail as they get further away from a blood vessel, and they're trying to compete. Oh, I want to be over there. There's more sugar, there's more oxygen over there. And they start fighting with each other. And they start having changes, as Dr. Meltzer discussed and Dr. Brown discussed. They start mutating, they start varying a little bit across the way. And they keep growing and growing and growing, but they're different. And some might be frontiersmen and women that are pushing the boundaries and expanding where it can go. Some may be even more daring explorers that jump across the body and try to set up in the brain or the bone or lungs or just lymph nodes and spread. Others just stay where they are and try to outcompete in these major cities and these crowded areas that run out of sugar and need to make new blood vessels and new roads and new ways to grow. But all of this fighting is happening way before the diagnosis. So when we say you have cancer, there are lots of cells, lots of animals, lots of variation. A 2-inch by 1-inch tumor has about 8 billion cells in it, about the same as the population of humans on the planet. And you can see from these tumors, they're way bigger than 2 by 1 inch. So billions and billions and billions of cells. So the problem is now we have a new species in our patient, and we want to eliminate it. As opposed to all the efforts you've heard where we don't want to make more animals extinct, cancer therapy is trying to make this new species go extinct. We're trying to cause extinction. So the problem is cancer is mutating even more, and so the diversity is even more in each individual patient's cancer than all of the humans on this planet. I don't want you to think about how to eliminate all the humans. We don't want to do that. But cancer is our most personal experience with evolution. Hard to imagine connecting us to dinosaurs that far ago. It happened, but how? And this cancer is evolving fast. Cancers are not dividing. I had my first kid when I was 28. Cancers are dividing daily. In some of the models and some of the cancers we've explored, like breast cancer and other cancers, the tumor that was there 10 days ago, none of the cells are still alive 10 days later, even as the tumor's bigger. So much change, so much not static, so dynamic. So it's fast, it's diverse. Is it too big a problem? No, it's not. We can cure many cancers. 60% of cancers are curable. But there are some more challenging ones that spread fast, ones that grow fast, ones that resist our therapy. Dr. Callan has nicely, for the past decade, said, so what? Like, what's the what? Where's the what to this approach? Which I appreciated because it sharpens the idea. And the what is, if it's an animal, if it's a new species, if we're trying to make it extinct, that is something we can study. It's a worldview where you can kind of explore things. 99% of creatures on this planet have gone extinct. Extinction happens all the time, all the time. So the so what is 98% necrosis or 98% tumor kill is not extinction. Shrinking something is not the same as eliminating something. And it could be that we have great drugs or therapies that can kill most of a tumor. And we might have some therapies that are great at killing the last bits of tumors. We'd have to think of those differently. You can't expect something that would kill the last bit to run through the gauntlet of drug development and shrink a tumor along the way, when it can't do that, but it can eliminate the last bit. You don't want to keep finding drugs that shrink tumors, but don't eliminate it, because then you're not making the cancer extinct. So that's one so what. Another good so what is that we typically give agents, shrink a tumor, and say, let's keep riding that until the disease comes back. We give away the advantage to cancer. We let cancer lead to progression, and then we try to catch up after it's relapsed. And so the so what would be to anticipate. An eco-evolutionary oncologist should anticipate resistance, study resistance, and change therapies. Is there any model for changing therapies? There definitely is. And so there is a pediatric cancer called acute lymphoblastic leukemia that was just as devastating as some of the metastatic solid tumors are now in the early 60s. And therapies are given, and after 6 weeks of those therapies working, we completely abandon them and change to other therapies. We do series of strikes. So you think of extinction and you think an asteroid hit the planet, volcano went off, some mass extinction event happened. And it's great for movies, but it's not how extinction happens. Most of the 99% of species that went extinct, and I'm happy you're nodding your head because it's good to have a fact checker. Most of the animals that go extinct do have a population reduction, but then a series of unfortunate events keep happening. And that's exactly how we treat childhood leukemia. 6 weeks of something to massively reduce the white blood cell count, and then change. And then 6 weeks later, change. And then 8 weeks later, change. And then after changing, and many months after all the disease is gone, 2 more years of therapy to just try to make sure nothing comes back. And so a series of changes, smart changes at the right time with the right agents. It was deduced in leukemia. We don't have enough, some solid tumor patients like osteosarcoma, to just empirically figure it out, but trying to learn when to change therapy and what to change it to is important. And then finally, Dr. Meltzer mentioned that with a lot of cancers, they kind of say, here's why I'm a cancer. Here's the gene that's up. Here's the gene that's mutated, and you can develop a drug to knock it down. So, a lot of cancers don't have that. And so evolution is selecting sometimes favorable mutations, but evolution is also tells you something when a gene is never mutated. If you have a cancer, you've sequenced hundreds of osteosarcomas, and if you look across all of those 300 plus patients, there's 82 regions that have never changed. That's important. While many people would say, boring, it doesn't change it, you can't target it. I think absolutely you could target that. Changing in either direction is not tolerable to cancers. And so things that don't change could become therapeutic targets is another way that I would think about things. So I agree with Dr. Meltzer. I think a good understanding of cancer and good therapeutic advances are coming. With the next slide, I have the pleasure, and it's easy to care about osteosarcoma. You just have to meet one patient, and many of us have met many of them and their families. And there are heroes, and some of these patients and families are in the room today. And I think it's fantastic that the petitioners organize this event and to think about cancer a little bit differently. Cancer is a fundamental problem of being a multicellular creature, which I enjoy being. And but having a good understanding of something as far off as dinosaurs to as personal as an experience with each individual cancer patient, I think has been a wonderful hour thus far, and I'll turn it back to Dr. Brown. Thank you.
All right. Well, I'd like to thank all the speakers. I think you've given a very good picture of the battle, the foe that we're fighting, and how it began. We'd like to take questions from the audience. Does anyone have any questions? Well, I can start by just asking Dr. Reed. So I think you alluded to this, but maybe we need to expand it. I mean, there are 2 models. There's a model you talked about childhood leukemias where there are multiple mutations and the tumor keeps evolving. But there are other cancers where there's a certain essential gene that causes the cancer, like chronic myelogenous leukemia in adults. So maybe you should contrast those 2 for the audience.
Good. And so it's tough to talk about cancer as one entity. There are hundreds of different types of cancer, and I think this is an excellent question. We'll pick 2 cancers. We'll pick chronic myelogenous leukemia is a good one where there's a quiet genome and one dominant change that kind of takes a cell from normal to cancerous. That dominant change has a protein that there's a binding pocket, and you can put a small molecule in there and block it. So 20 years ago, and one of the reasons I went into medicine was this was happening. You just figure out what's the Achilles heel of every cancer, what's the drug that fits in that, and let's eliminate it. There are cancers in the solid tumor realm, like GIST, that have a similar kind of pocket that you can fix. Focusing on the cancer that we're caring a lot about today, osteosarcoma, there isn't a pocket. And the recurrent changes and mistakes and mutations in P53 are these massive proteins. If we're talking about a small molecule, it's now the whole room, this big protein that does all of these tasks. And you can't put that in a pill and swallow it or inject it and replace that protein. What's missing is so much stuff that's so fundamental to so much biology, and so we don't have a druggable solution for that. And so there are some cancers, Dr. Hellman uses the word smart and dumb cancers. He would say the targetable one is dumb. It's kind of like we're smart enough to figure it out already. But the cancers that are rather complex, that don't have a clear target, that are changing faster than we can put it on a wax and put it through a sequencer and figure out all the DNA, but they keep changing. Those ones are much more complicated. And I agree, for a cancer that's very curable, that has a drug with little side effects that cures it most of the time, you don't need to go through the machinations of thinking about evolution and all of this sort of things. But for the ones that we don't have a lot of insight, it is, I think, worth bringing in other ideas.
So, nobody wants to ask a question? Have we been so cogent? Have we been, that you all understand everything? Or are you just intimidated?
I've got
Are you
one.
like students?
I take it by virtue of the degree of excessive mutation that this particular cancer has, that the virus treatments that are out there would not necessarily be effective. Is that correct?
I could do it.
Well, that has been the track record. But I would say that there's active research. There are those who are believers that it's still a possibility to find, because those kinds of viruses that are working well in leukemia, is that sort of what you're referring to? They target a specific protein, and there may be specific proteins that could be targeted. So far, the use of those kind of viruses against any solid tumor has not been super encouraging, although there are some glimmers of success here and there. So it's well worth thinking about. The kind of therapies that you also read about and get headline news that are immune checkpoint inhibitors that really have transformed aspects of adult oncology in particular, so far are not very active in pediatric tumors. But again, there's room for further research to really pin this down. And I, for one, think it would be wonderful if we could figure out how to use those tools in this disease. And I think certainly my personal evolution has been from trying to find that magic thing like the CML target that you would give a pill and make a big difference, to anything, whatever it might be that works. We want it.
Thank you all so much for all you're doing in so many realms of this that affects all of us on this planet throughout history. I just wonder, what are some of the biggest barriers you find to your research? And once you find those magical targets, to being able to take them forward, what barriers keep you from being able to plow forth?
I think that the medical establishment, clinical trials, scientific research and inquiry, and all of that stuff is perfect for this magic bullet idea. If it's a virus or anything, if any of that happens, we're all set. So we're all hopeful, eager, optimistic that we will just discover that in each cancer. We heard that in osteosarcoma 40 years ago, advances, and not much since. With metastatic solid tumors, that's true almost everywhere. I mean, immunotherapy has been a game changer for some adult tumors. Again, it's hard to talk about all cancers at once. But largely, if you have a solid tumor that's spread, there isn't the magic bullet, though. And so I do hope that people keep picking away cancers one magic bullet at a time. But as an evolutionary oncologist, I think that the lessons are that we need to do more than one thing. So the barrier that I personally feel is trying to change enough hearts and minds of my colleagues so that we could study more than one intervention at once. I fundamentally don't think we're one magic bullet away from big changes in some of the more complex cancers.
Are these cancers more prevalent now, the sarcomas?
I'm not an epidemiologist, but I don't think sarcomas have gone up dramatically in terms of epidemiology. I don't think that osteosarcoma is vastly more, they're not lifestyle diseases, is what I would say. The cancers that have changed because of diet or obesity or smoking or sun or other things like that, these aren't those cancers. So I think their epidemiology has been relatively stable.
If you look at cancer statistics across America for the last decade or two, there's about 1,000 new osteosarcoma diagnoses each year in the United States. And so it's incredibly rare. And as Damon said, not a lifestyle cancer. We're not finding it more. We're not seeing an increased incidence. Right. It's just bad luck.
Right, and if I could make one comment that's slightly tangential but relevant to one interpretation of the question, is that the thing that has changed tremendously in the last several years is the correct precision molecular diagnosis of sarcomas. We now have taken groups that had one name and split them into several really different diseases that will have really different biology and which are being individually researched. And that has had a huge impact on diagnosis, less so on treatment so far, but in some cases it is starting to affect treatment. So we have a much better understanding of the different varieties of these diseases.
But there are common mutations. For example, colon cancer. Essentially all of them have mutations in one particular gene. Now they may evolve beyond that. They do evolve beyond that and have other things. But don't you think that there's a common initiating factor in certain kinds of tumors? You find the same mutations recurrently in tumors of a particular type. So that suggests that, that the cancer really does need that. Now, maybe, as Dr. Reed, as you say, maybe it evolves then and it doesn't need it anymore. But there appear to be two kinds of mutations, some of which the tumor is addicted to, and if you can get rid of that, the cell will die, and others in which the mutation kind of helps the tumor, but it's not essential. So you're right, it's extremely complex and it's continually evolving. But the dream of every scientist is to find the one thing that the tumor really needs, where if you could get rid of that, you would get rid of it and all of its relatives. Speaking of relatives, I wanted to ask Dr. Callan, you mentioned that osteosarcoma is more frequent in certain species, in certain breeds of dogs. Do we know what's different about them, or does this give us an opportunity by comparing, sequencing those breeds to find mutations?
So I think at the simple level, it's large and giant breed dogs that develop cancer more frequently than small breed dogs, and the genetic difference is a difference in one gene that regulates bone growth.
Right. So there are genes that can explain, or at least partially explain, why one species is more, one breed is more susceptible than others. Well, that might be a clue to people too.
Now, just quickly add that just to answer the question, osteosarcoma dogs is somewhere between 10 and 100 times more common than in humans, but not changing.
They want the room back, so thank you all for coming. There is a reception upstairs and be an opportunity to visit with the the panelists and with each other and have a glass of wine or whatever. So thank you again for coming.